Okay, so just going to move a little bit further out into the ocean, although this project very much overlaps the other projects, so you'll see some similarities. I'd just like to acknowledge all the other contributors to this work. So I'm presenting mostly what I haven't done by the relatively large team from CSIRO and AIMS and around the place. I'll try and give you a quick summary of two different scales of biogeochemistry that are of interest to us. Of course, biogeochemistry is sort of a complicated word, but it's really about productivity, right? So it's what fuels primary productivity, and then of that primary productivity, how does that start making its way up into the food chain and then into things that you've been talking about over the last day and a half. So the aim of this project was really at two scales, shelf scale dynamics, and this was really the work of Jim Greenwood at CSIRO and his team. And then the base scale biogeochemistry, which you've heard a little bit about from Nicole just now, and I'll talk a little bit more about how those two overlap. In terms of modelling scope, the two models you're going to hear about now are the one you just heard about from Nicole. It's the same model, same package. We're going to look at it through a slightly different lens. And then I'm going to talk to you about the larger scale shelf model. Before I do that, I just want to highlight where some of the data comes from, because actually one of the best contributors, I think, of this project was actually just pulling together a whole bunch of different data sets. And I just want to acknowledge first the first cruise that was collecting biogeochemical data in 2010 by the Southern Surveyor, led by Peter Thompson and his colleagues, and really just creating these data sets of transects off the shelf there and providing some benchmark data that we've been able to build upon. The picture there I should just show you is phytoplankton abundance, chlorophyll A, and you can see the variability as you move near the coast and then start moving offshore off the shelf. So if we look at a slice now, sorry, this slice is backwards, but you'll just have to imagine. You can see a slice through this data. You can see this just gives you a general picture about what the shelf scale biogeochemistry looks like. First, oxygen here, the well oxygenated layer, low oxygen zone as you progress off the shelf edge. Temperature as well, stratified there, and then transmissivity is basically water clarity, so you can see the extreme high turbidities, very high up here, and then dropping off as you go deeper, as you would expect. If you look at nutrients now, and we switch back so you can put your brains back the right way. Nitrate, here you can see deep nitrate coming up the shelf edge and then depleted in the surface. Total chlorophyll A, high chlorophyll near the edge of the coast, and then also a high bit around there where the gradients occur, silicate, phosphate, and ammonia. So that gives you a broad picture of what's happening in the region. So the next question is, what is going on in terms of the productivity? What controls productivity? How do those nutrients move around and get used? So one of the first bits of work that was done was really analysing some of the productivity data from Miles and his colleagues. And this shows you two different places. And this was done all over the place, but I'm just highlighting two. One here is a shallow water station where what you can see on this side is productivity, so the amount of photosynthesis, the amount of carbon that's fixed versus the amount of light that is received. And what this shows is that these guys, they like as much light as they can get. You can give them more and more light and they keep photosynthesizing. Whereas on the other side here, we have a photosynthesis curve that shows that has a peak at very low light levels, and then they actually get what we call photo-inhibited at high light levels. And this is typical of when you get a deep chlorophyll maximum. So the phytoplankton have adapted. They're used to photosynthesizing in the low light areas where there's relatively high nutrients, the nutricline. So you can sort of think of these, just for those that aren't into phytoplankton. I've got this guy here. He likes the sun, and this one is like the vampire plankton. They don't like the sun. They like to be in relatively low light. So if you compile some of that data, what you can see is that as you get deeper, the chlorophyll goes down, but there is this sort of peak on the edge. And similarly for nitrate, you're seeing this sort of trend here as well. So now I want to take you to the model. So this is now the 3D model, ROMS model with a tracer in it. This is, again, this is work by Jim and the team at CSIRO. What they did is they put a tracer of nutrients, so this is essentially like nitrate, below the nutricline, and then they start the simulation like this, and then they run the simulation for 1 year. And slowly, because of all the mixing and dynamics that are happening across that region, you start getting this upwelling of nutrients into the surface layer. And then we want to see how are those nutrients going to be propagated through the domain. Of course, they're going to be driving productivity. And what you can see here is after 1 year of simulation, the map of the tracer that has come up from the bottom water to the surface. You can see it very clearly along the shelf edge there. So that gives us this interesting story that the upwelling caused by internal waves and so on at the shelf edge are mixing up and fueling productivity. And if you compare the model with the data, you can sort of see here, this is sort of some averages. This is a model looking at chlorophyll. This is nitrate. You can see the nitrogen getting higher as you go deeper, and the model seems to be capturing that. This is the low maxima in chlorophyll, and little bit over predicted by the model. So it's here sitting around 30 meters instead of maybe around 60 meters. Sorry, about 40 meters instead of about 60 meters. So there's some issues there in the vertical structure, but the shape is reasonable. So then the model is run with a biogeochemical model. It's a bit simpler than the one that Nicole described, but it has nutrients and phytoplankton and zooplankton grazing. So it's telling us the base of the food web. And what we see, this is those observations I just showed you, but what we see in the model is we over-predict how much is happening in where the shelf is, and then we under-predict at the coast. And there's a good reason for this. This was actually sort of an interesting story because the model was set up only to have those plankton that were on the right side of the diagram before. So the shelf model was set up to have photoinhibited plankton. So it's not surprising that they started to die when we got close to the coast because it's just too sunny there for them. And of course that leads us to an issue of scale where the particular model setup was working well off this side of the coast, maybe over predicting here because of some of the simplicity of the biogeochemical microbial pathways, and then under predicting here because of that assumption about their sensitivity to light. So now we move down to the bay scale and try and answer that question a little bit more. So there was 2 cruises. Greg's told you about those cruises that were on the Solander in 2013, which was a dry part of the year, and then in 2014, which was a wet part of the year. Same transects. There was a whole bunch of data collected in that, and I just don't have time to go through it all, but ranging from nutrients and right through to cell counts, cytometry, and other measurements. So here's an example now of just a vision of the model, same bay as you just saw. This is Walcott Inlet here. Now we're in the bay. These dots are the transects that were taken on those cruises, and there's the Montgomery Reef. So if we now try and conceptualize, we're going to think a little bit more, I guess, about the next step from what Nicole just described to you. We're trying to understand a little bit more about redistribution, recycling, primary productivity. How does light control photosynthesis? Remember, it's a very turbid environment. There's a lot of mixing, so Greg just told us about all of this stuff coming in and out at a rate of knots. We've got a general pathway going through here. We've just learned as well from the large-scale modelling that there's upwelling of nutrients that are coming into the system. And so we've got this story which is a combination of transport and fate, which is controlling primary production. So we go back to the model, the same model as you just heard about. The model was built on a reasonably sophisticated data set collected by the team. So here's an example of the chlorophyll in different size fractions compiled by Miles Furness at Ames, and then zooplankton size fractions as well here, looking at different bays, including Collier Bay by Dave McKinnon as well. So it's a nice, very nice data set there to help basically help us conceptualize this system. So the model was used to predict, you saw some outputs from that already, including the suspended solids and how that was changing in that diagram from Nicole. And on top of that, we can start to look at how productivity was varying within the embayment too. And what we were interested to see here was that we actually have relatively patchy emergence of high areas of phytoplankton chlorophyll primary productivity. And so on this side is a dry season, on this side is the wet season. And what you can see is that there is sort of a tendency for high productivity near the coast. And that is using the other type of plankton that I showed you in the early one, which don't become photoinhibited. So it's sort of a little bit paradoxical, because you think that it's so turbid, there's not much light. But what's actually happening is the phytoplankton are extremely efficient at photosynthesizing in their excursion as they're moving up and down through the water column created by all this mixing. So here's another similar okay, so it's not going to play, but you saw Nicole's earlier diagram. The one thing I did want to show you, which is neat about this animation though, was this is not showing you nutrients moving through the system, but it's showing you what we call the light limitation function and the nutrient limitation function. And so what you see here is, even though it's really turbid, you get this sort of step change from the inner to the outer region, where the light limitation is able to penetrate just that bit deeper as you go into the bay. But the light penetration is very thin here, but it's enough for them because there's such a high vertical diffusivity that they keep getting exposed to light. And it's a little bit different. We have two main groups there, diatoms and phytoplankton. Whereas the nutrient limitation, however, is more horizontal. So you see light limitation vertically with this sort of step change, and then this sort of gradient of nutrient limitation as you're moving out. So that helped us conceptualize the controls on productivity. What is the importance of light, sediment, turbidity versus nutrient supply from the terrestrial sphere? On top of that, there was a whole bunch of empirical work done by David McKinnon, including not only the two cruises that I described, but he also compiled his earlier work from 2011 across a whole bunch of sites. And what he showed was not only the size fractions of zooplankton and compiled them across the region, but also their relative activity. And he did some really novel measurements looking at the growth rates or proxies of the productivity of zooplankton. So in other words, how fast are they growing and how fast are they respiring? And this is critical. This is the link between primary productivity and the higher food web. And so this is a really novel data set that is able to help answer that question. What he shows here is these two metrics for different sizes of plankton. Those different sizes of plankton, of course, have different roles in the food web. The other thing he did that was quite interesting is compare this to Great Barrier Reef samples where he's done very similar work. And one of the questions we had at the start of this was, well, how similar is the Kimberley, or how different is it to other systems that have been well studied? And so this was quite interesting to see that despite it being very turbid, and you would think maybe not as productive as other places, well, we're seeing that there is productivity there. It's reasonable, it's comparable with other areas, but also the secondary productivity, so zooplankton going up, is actually higher. It came to be 4 times higher in his average measurements of all those sites than his equivalent data set in Great Barrier Reef. And there was a higher productivity on average. And using this is all empirical data, this is not the model. He estimated around 7% of zooplankton, 7% of the primary production went up into zooplankton. And so that's also equivalent to the numbers we used in the model, that Nicole showed as well. So there's some interesting stories there about the controls on primary production, but also the significance of secondary producers taking that up. So the last summary there, I've got the bell. Tidally driven upwelling of deep nutrients was creating a chlorophyll high on the shelf edge. Within the 50-meter contour, very high velocities and vertical mixing create sort of a complex controls on primary productivity. Despite the high turbidity and very high light attenuating conditions, we still actually had a relatively significant primary productivity that corresponded with those phytoplankton that were more adapted to that region and not photoinhibited. And finally, the secondary production was also an interesting part of the story in transferring that carbon up the food web. Thank you very much.
Thanks, Matt.